Perfect. So this diagram is essentially the grand family tree of gram-positive bacteria, and we'll explain it one by one using our microbiology algorithm that will basically simplify all the jargon. So let's make our own algorithm here. Now, big picture is you first ask, is it gram positive? How do you know something is gram positive? You know something is gram positive when it is basically purple or blue. So hey, can you see that the gram-positive bacteria is blue and gram-negative bacteria is pink? Why does this happen? Because gram-positive bacteria basically have a thick peptidoglycan wall. So this thick peptidoglycan wall basically retains the crystal violet dye, right? And that makes it purple. Now, after you know that something is gram-positive, you further on go and see whether it's a bacilli, cocci, or does it have branching filaments. So if it's a bacilli, we call it a rod. A rod is basically a bacillus. And if it's a sphere, if it's like circular, we call it cocci. And if it's branching, then we call it branching filaments, right? So moving on, I'm going to show you these three pictures here. So this is basically a gram-positive bacteria with branching filaments. This is a gram-positive cocci, which is in clusters. You can see they're spherical and circular. And then this is a gram-positive bacilli, which is basically rod-like. Right. Okay. So let's first study the bacilli, which are basically aerobic or anaerobic. Now, in order to study the bacilli, let me take you. When we come to looking at bacilli, basically, we'll first look at Listeria. Now, this is an aerobic bacilli, right? This is a gram-positive rod because it's a bacilli, we'll call it a rod, and it's facultative intracellular. When we say something is facultative intracellular, it basically means it can hide inside cells, right? Now, I'll go and show you Listeria. This is how Listeria basically looks like. Now, Listeria, basically, I said it's a gram-positive rod. Now, you can see these are all rods. They are bacilli. And they're facultatively intracellular, so they can hide inside cells. Now, if we talk about Listeria here, basically, the special thing about Listeria is that it has actin rockets. These actin rockets basically hijack your actin, right? The actin is basically the normal molecule that helps you move your cells. So Listeria says, thank you, I'll use your actin rockets. Then it shoots itself from one cell to the next cell. So cell A's Listeria uses an actin rocket to shoot to cell B, right? And this intracellular movement, this basically actin polymerization allows intracellular movement and cell spreading across cell membranes. And what this basically does is this avoids the antibody. And you see this characteristic tumbling motility in the broth of Listeria. All right. So it's a gram-positive facultative intracellular rod. It's acquired by ingestion of unpasteurized dairy products and cold deli meats. There's transplacental transmission also. So while the baby is in the womb, the mother can pass it to the baby, and by vaginal transmission during birth. It grows well at refrigeration temperatures in a cold environment. Now, diseases of Listeria include neonatal meningitis. Pregnant women eat the unpasteurized milk or soft cheese, and the baby develops meningitis. And then the special thing about meningitis here is that meningitis is especially seen in Listeria. It's seen in neonates, it's seen in the elderly, and it's seen in immunocompromised patients. Then this also causes gastroenteritis, which is food poisoning in healthy people, and it's treated by ampicillin. And sometimes you also give ampicillin plus gentamicin. So remember that a buzzword to remember Listeria would be cold growth. So it's basically grown in refrigerator, right? So you'll see it in unpasteurized milk and dairy products, right? Um, so when we saw bacilli, we saw the aerobic bacilli, right? So we've done Listeria. Now we will look at bacillus, right? In order to look at bacillus, look at this picture. So this is basically bacillus. Bacillus are large aerobic spore-forming rods, and they have basically two important species. They have a Bacillus anthracis, and then they have a Bacillus cereus. So first, let's look at the Bacillus anthracis. It's a very dangerous kind of a bacteria because this toxin of Bacillus has three further components. It has a protective antigen, which is basically the key. And like a key opens a lock, basically the protective antigen allows the bacillus to enter the cells. All right. And then it also has an edema factor, which basically raises the CAMP. And since the CAMP gets risen, there's edema. So you always get edema around this anthrax scar. There's always edema around this because of this edema factor. And then it also has the lethal factor, which basically kills the cells. So this protective antigen helps it enter the cells, helps the toxin enter the cell. The edema factor basically allows for all of this swelling around the eschar, and the lethal factor basically kills the cells. Okay, so you get this black eschar in the disease, which basically means black skin, dead black skin. It basically it looks like an insect bite look, like it has a black center. So it's a painless papule surrounded by vesicles. It's an ulcer with black eschar. It's painless and necrotic, and it's uncommonly progresses to bacteremia and death. Then you have something called the pulmonary anthrax. That is basically inhaled spores. Since it's a spore-forming bacteria, the spores are inhaled, and that basically causes a severe lung disease, right? One thing that I would like to point out is that the Bacillus anthracis, basically, something to remember about this is that the capsule of anthrax is a little different in the form that the anthrax capsule is made of poly-D-glutamate. This poly-D-glutamate basically, what happens is it allows the bacteria to evade phagocytosis. Okay, and transmission usually comes from cattle, sheep, or goats, right? So exposure occurs through animal hides or wool, fur, meat, or spores in the soil. And the spores can survive for decades, I must mention it here. Why are these spores important? We're talking about pulmonary anthrax, so it's important to see why these spores are important. Basically, think of the spores as sleeping armored bacteria. So it's a whole armored weapon driven bacteria, but it's sleeping. So they can survive heat, drying, chemicals, and harsh environments. And when they enter the body, they basically wake up and become active bacteria and cause the disease. So, let me get to the pulmonary part. Yes. So what happens is the spores enter the lungs and the macrophages basically eat the spores. And when the spores are eaten up by the macrophages, the spores germinate. They basically multiply and they travel to mediastinal lymph nodes. When they travel to mediastinal lymph nodes, they cause a massive hemorrhagic inflammation over there and cause basically mediastinitis. Mediastinitis is basically inflammation over there, right? Massive hemorrhagic inflammation. And this leads to shock and this leads to death. So that's something you would want to remember. And this is basically known as the wool sorter's disease, right? I told you it's in sheep. So this is how the spores enter the lung. I should classically make this here. Okay, so that's your pulmonary anthrax. Moving on to the other kind of bacillus, which is Bacillus cereus. The Bacillus cereus is special because now Bacillus cereus is closely related to Bacillus anthracis and Bacillus subtilis, which is also another type of bacillus, which is not important. But remember that unlike anthrax, the Bacillus cereus mainly causes food poisoning. Again, it's a gram-positive rod. It causes food poisoning, and it the shape is like a rod since it's a bacillus. And yes, it is spore forming. The spores survive when you're cooking rice. The spores basically survive, and when you reheat the rice, that's when these spores basically become the bacteria. And yes, they're motile, and their habitat is basically soil, dust, and food. So the usual transmission is through contaminated food. You always get this Bacillus cereus through reheated fried rice. This is why it also is called as like the fried rice syndrome. Okay, why rice? Because when rice is cooked, most bacteria die, but the spores are surviving. And when the rice is left warm for hours, after that, these spores germinate and the active bacteria multiply and toxin is produced. The virulence factors are important. There are two virulence factors here. The first one is the emetic syndrome. So you can see this emetic type. The emetic type is basically emesis means what? It means vomiting, nausea and vomiting. And there's a heat stable preformed toxin, and we call this the cereulide. Okay, I'll write that here. Basically, the type 1 is caused by cereulide. That's a heat stable preformed toxin. What does the preformed toxin mean? The food is sitting out, bacteria grow, and the toxin is already produced. When you eat the food, you get the immediate symptoms. The onset is 1 to 5 hours, and this is very easily tested. And the symptoms is basically nausea, vomiting, minimal diarrhea. I ate rice and started vomiting a few hours later. The second thing is the diarrheal type. Now, the diarrheal type is a different toxin because it is produced inside the intestine after ingestion, right? So you don't get it very quickly. The other one you get within 1 to 5 hours of ingestion. So the diarrheal type, you eat the contaminated food and the bacteria reach the intestine, and that's when the toxin is produced in the gut, and fluid secretion happens, and you get watery diarrhea. Right, timing of the other one is basically 8 to 18 hours. It's later than the vomiting syndrome. What do you get? You get watery diarrheas, abdominal cramp, and GI disturbance, GI pain. Right, so the second one is 8 to 18 hours. Usually there's no blood in the Bacillus cereus watery diarrhea. So why the different timing? Because in the emetic time, you're having this preformed toxin consumption. But in the diarrheal type, the toxin is produced after you intake the heated rice. So we've done the Listeria, we've done the Bacillus, and now we're moving on to the Corynebacterium. Okay. We now move on to Corynebacterium diphtheriae. So Corynebacterium diphtheria, as I said, it's a bacterium which is gram-positive, and the shape is again like a bacillus. It's a club-shaped rod. The arrangements are like Chinese letters, V-shaped and L-shaped. So, and they're spore forming? No. So these bacillus rods are not spore forming. The shape you can see they're either L-shaped or V-shaped, right? So these are V-shaped and these are L-shaped. And they're not spore forming, they do not have a capsule, and they basically cause diphtheria. Diphtheria is this why is it called Corynebacterium? Corny, basically this corny word means club, right? And the bacteria basically looks like little clubs. Instead of straight rods, they often appear at angles. And the bacteria divide, but they don't completely separate, right? They form these Chinese letter arrangements, as you can see here, V, L, Y kind of arrangements. And that is something to remember. And by the way, exams love this appearance, this VLY shaped. The examples are the spread through respiratory droplets. So examples are coughing, sneezing, or close contact. The virulence factor, this disease is not caused by the bacteria itself. The damage is caused by the diphtheria exotoxin. This is very important. So the exotoxin is the problem in this, is the viral factor, virulence factor in Corynebacterium diphtheria. Now, where does this toxin come from? The toxin gene is carried by a beta prophage. A bacteriophage virus infects the bacterium. The virus infects the bacterium, adds the toxin genes, and the bacteria itself becomes dangerous. And that's what beta prophage does. Let me write that down here and we'll explain it further. So the beta prophage is a bacteriophage virus that infects the bacterium. And what happens is that you add this toxin gene and the bacterium becomes dangerous, right? And now without this beta phage, there's no toxin and there's no severe diphtheria. So mechanism of the toxin is one of the highest yield microbiology concepts that you must get here. This toxin causes ADP ribosylation of elongation factor 2. Okay. During protein synthesis, if you remember, the DNA becomes the RNA and the mRNA becomes the ribosome, and then that becomes the protein. Now, the elongation factor 2 acts like a conveyor belt and it moves the growing protein chain along the ribosome, if you remember. Now, what happens is this toxin destroys that conveyor belt of the elongation factor 2. Since that elongation factor 2 is blocked, the protein synthesis stops and the cell dies. So since there are no proteins, there's no survival, right? So A, B, C, D, E, F, G, A, B, D, C, A, B, C, D, E, G remembers. It will help you remember epiribosylation, which is basically um, uh, happens because of the elongation factor, and basically the protein cannot be synthesized. And the B is for the beta prophage, which infects the bacterium and adds the toxin gene. And C is for Corynebacterium, D is for diphtheria, E and F is for elongation factor 2, and G is for granules. Um, Diseases caused, it basically affects the throat, heart, and nervous system. The bacteria basically colonize the throat, the toxin kills the local cells, and dead tissue accumulates, a large membrane forms, and airway obstruction may occur. I've only seen this case once in an underdeveloped setting. That's the only time I ever saw this case, but it was very um, easy to pick it because it's almost impossible to miss. It's a famous pseudomembrane. There's a gray-white pseudomembrane, and it's impossible to miss. I'll show you one here. So this is how that gray-white pseudomembrane looks like. It's almost impossible to miss. Um, what else can I tell you about this? The pseudomembrane, remember that the pseudomembrane itself, it's made of dead cells, fibrin, inflammatory cells, and bacteria. Why is it dangerous? Because, oh, remember, whenever you try to remove the pseudomembrane of Corynebacterial diphtheria, it bleeds. Why does it bleed? Because it's very tightly attached. Local filter symptoms will include fever, sore throat, difficult swallowing, and you see this gray-white membrane. And large cervical lymph nodes may cause a bull neck. So you may see a bull neck appearance. The systemic toxin effects will include the heart. So the heart gets myocarditis because of inflammation of heart muscle. The nervous system will show you neuropathy because the nerve damage causes weakness, paralysis. Remember that a classic examination case will be like an unvaccinated child with sore throat and gray white membrane that bleeds when scraped, and that is your diagnostic clue for Corynebacterium diphtheria. So this is your diagnostic clue. How will you diagnose it? You'll get gram-positive rods in Chinese letter arrangements. You'll get metachromatic granules. These are seen on Loeffler's medium. This is also very commonly tested. You get these metachromatic granules on Loeffler medium. And what are they? They are basically storage granules containing phosphate, and they stain differently. They appear blue and red in the same bacteria. So that is something that helps you diagnose them. These are the metachromatic granules of Corynebacterium diphtheria. They appear blue and red. Here you can see them. They're not red here, they're blue and green, but usually they stain differently and they're called meta because they change their color. Why they call metachromatic? Because they change their color. They stain a different color than expected. It's also grown in cystine tellurite agar. It's mentioned that it produces black colonies. Why? Because the organism reduces telluride. I guess that's enough for you. Let me see the tests that are mentioned. Oh, there's also another test that I should mention, the ELEC test. There's an ELEC test also. So what does the ELEC test do? It's usually used to determine if a toxin is present. So positive ELEC test will basically tell you if there's a toxigenic strain, right? So a Corynebacterium diphtheria weak toxin is positive, and a strong toxin is also positive, and then this one is negative. So these tests basically tell you if the toxin is present or not. An immunoprecipitation line forms where the diphtheria antitoxin meets from this disc meets diphtheria toxin from an organism. So it's used to determine if the toxin is present. I want to show you the cystine telluride blood agar. Also, this is mentioned. It produces these black colonies. Why? Because the organism basically reduces tellurides. Treatment is very important. You must do two things. You need to kill the bacteria, and you kill the bacteria usually by erythromycin and penicillin. That is something to remember. You neutralize the toxin. How do you neutralize the toxin? By basically giving a antitoxin agent, right? So you give erythromycin and penicillin to kill the bacteria, and you neutralize the toxin by giving a diphtheria antitoxin because antibiotics themselves cannot remove the already circulating toxin. And prevention is by the DTaP vaccine. The diphtheria toxoid is a toxin which is inactivated and it cannot cause disease, but it still stimulates the immunity, and that's why we give the DTaP disease. I'll stop the video here because it's getting too long.